All right, you guys, welcome back. Here we are in chapter 13, which is all about the chromosomes. Now, we've been talking a lot about chromosomes, a bit in each chapter. I mean, we talk about how they are segments of the DNA that's coiled up, how genes are located on the chromosomes, how the chromosomes move and separate during mitosis and meiosis. But now we're going to talk a little bit more about them. All right, so in this presentation, we're actually going to talk about sections 1 and 2. So as you can see, we have 1 and a 2. And are you guys ready? Yeah, so let's go. Woohoo! All right, so there it is. All right, let me adjust this. There we go. So, portrait of a chromosome, or as I like to call them, the chromosomes. No, that is not a technical term. That is strictly a Komar term. So, we have this thing called cytogenetics. Now, we all know what genetics is. After all, that's what this class is. But cyto refers to cell. So cytogenetics is looking at some of the things that are going on in the cell, specifically the genetics of the cell, and where we find our genetics, the chromosomes. So cytogenetics is a study of chromosome abnormalities and associated effects on health or other traits. We've talked about mutations in genes, but there's also chromosomal mutations, and they don't just affect one gene, they can affect multiple genes. Excess or deficient genetic material can cause syndromes or end prenatal development. So there is a wide variety of different syndromes, disorders that can occur if you have too much genetic material, meaning you have extra chromosomes or extra pieces of chromosomes, or if you have too few. So if you're missing a chromosome, because as you remember, they all come in pairs, but what if you only have one? And what can happen then? Or if you're missing a section of a chromosome. So you can lead to, at the very least, different disorders. And depending, And depending on which actual chromosome is the one with the abnormality, it can also end up or lead to a spontaneous abortion, which is essentially it's very similar to a miscarriage. So anyways, let's move on, shall we? Let's talk a little bit about the parts of a chromosome. So, chromosomes consist of highly coiled DNA, RNA, histones, and non-histone proteins. We've talked about a lot of these things in the past. So here's your DNA, wonderful double helix. So we have our nucleotides, our sugars, our phosphates, our bases, and then they wrap around histone proteins. That helps them to coil and squeeze down. Well, when you have a bunch of the histone wrapped segments, so we have those here in the picture, multiple histones, each with DNA wrapped around them, and then they're kind of coiled together. We call that a nucleosome. And then we take a bunch of nucleosomes and we kind of coil them together. And then we take those coils and we coil them again, and that's where we get the supercoils. And then that's essentially what the chromosomes are. Let's see. So let's move on. Telomeres. Telomeres are at the ends of the chromosomes, and they're repeated sequences, and it's believed that they are there to help protect the genes that are located on the chromosome. So if you notice here on the picture, this is a chromosome, and the darker ends, the darker ends, that's where the telomeres are. Now, what happens during cell division, and specifically DNA replication, the polymerase can't get all the way to the end of the chromosome. It can only build so far. So each time it goes through DNA replication and division, you lose a small piece of the telomeres. Now, it's this buffer region. It doesn't have any genes in it. It's just this repeated sequence. So if it's lost, it doesn't harm the organism. It's not going to affect any genes that are, essential for life functions. But they do decrease in size after every cell division. They're essentially just lost. They don't come back. And when a chromosome goes through cell division and it uses up the last of its telomere, it will not divide after that point. Once it's all done, it'll actually go to apoptosis, which is programmed cell death, and it'll commit cell suicide. And the reason for that is because if it was to go through DNA replication and cell division again, at that point it has no more telomeres left to lose, so it would actually start losing parts of genes, and that can be very, very detrimental depending on what genes they are. Centromeres. We've talked about centromeres before. Yeah, we have. Anytime we talked about cell division, whether it's mitosis or meiosis, we've talked about them. So if you look at the picture here, we have the chromosome, which is consisting of 2 sister chromatids. It's already gone through DNA replication, which is why we have 2 of them. And we always show that little part where they're connected, where the sister chromatids are connected together. That's the centromere. So a centromere is a constricted site where spindle fibers attach during cell division. And we've seen that in previous chapters before, how they attach to this region right here, and during mitosis or meiosis too, when the spindle fibers pull on the sister chromatids, it separates the 2 sister chromatids at the centromere and drags them to opposite ends of the cell. So centromeres are regions of repeated DNA bound to centromere-associated proteins. So again, we have these repeating sequences here. No genes are specifically located there because the point of them is to help connect their duplicated buddy and to also allow for a connection point, an anchor point with the spindle fibers. So there's proteins and repeated DNA sequences. All right, karyotypes. We've talked about these before. This is not new. We're just going to get into a little bit more detail. So, karyotypes are charts that display chromosomes in size order. Chromosomes are numbered from the largest to the smallest, so we do organize them by size. So, pair 1 would be the biggest, all the way down to pair 22, which is the smallest. And before anyone starts being like, Ms. Komar, we have 23 chromosome pairs. Yes, you're right. We always put the last one, the 23rd pair, as the sex chromosomes. Now, they're not organized by size. They just go automatically at the end. So it's 22, the first 22, the autosomes are organized by size, and then sex chromosomes at the end. So how do we distinguish them? Well, we mentioned by size, also the centromere location. Sometimes that centromere region is closer to the middle of the chromosome, sometimes it's closer to the end. We also look at differential staining. So if you've seen pictures of karyotypes, you'll notice that there are these dark banded patterns in them. And what those are, it's a particular dye that's used, a stain, that is picked up by certain proteins. Now, if it's 2 of the same protein, because remember your DNA comes in pairs, then they should have, so 2 of the same chromosomes, not proteins, sorry. So 2 of the same chromosomes, they should have, since they have the same genes, they should have certain proteins in the same location as each other. So that's why we can use them to help pair up the different chromosomes. We got some more information, like a picture. So here, I don't even know why I'm so tired. So here we have an example of a karyotype. So we have 1 all the way down to 22 in pairs from biggest to smallest, and then the sex chromosomes are at the end. Now, I can look at this and I can figure out some information. Oh, before we do that, sorry. You can, besides the size, if you look here at the first pair, notice how they have dark patterns in the similar locations, and other pairs have dark patterns in different locations. So that's another way. We mentioned the banding patterns. DNA probes, we're not going to get too much into that. You can actually attach something to the DNA and watch it kind of find it, have it go through division, or send in a radioactive probe, it'll attach to a gene, and then you can look to see where that gene is located. We're not going to worry too much about that. Anyways, I digress. Centromere location is a little harder to tell on something like this, or a karyotype like this. A lot of times what they'll also do is take the cell during either late prophase or metaphase when the sister chromatids are still lined up. Because when they're still lined up, it's very easy to see the centromere location. Once they're separate, it's a little bit hard. But again, looking at number 1, you can kind of see how it indents there a little bit. Same right there. That would be the centromere location. Notice it's kind of in the middle of the chromosomes. As opposed to this one here, you see a little indentation. We're at chromosome pair 4, closer to the top. All right, so a couple things we can tell from this. Now, karyotypes can give us a lot of information, but they cannot tell us about gene mutations because we cannot see what's going on in the individual genes at this point. We're looking at the entire chromosome. So it can tell us about chromosomal abnormalities. So it can tell us if any of them are misshapen, and before anyone's like, yeah, number 2 is death, and look at, no, I mean, we've seen DNA, we've extracted it from strawberries. You know it's a flexible structure. That's all that these bends are showing. But what we mean by misshapen is, are they fully formed into a ring? Does it have pieces sticking out? Is it cut too short? Things like that. We can also look to see in terms of if there's too many or too few. Now, when we did this, we looked through, we got 22 pairs of autosomes, no more, no less. We don't have 3 of one, like let's say a third 21st chromosome, which would lead to Down syndrome. We're not missing one of the sex chromosomes. So we aren't got that, we're good. So we have normal chromosomes, a normal karyotype, and we can also tell the sex of the individual. So if we look at the last pair, the X and Y, I know it's an X and Y because they ain't the same size. We've talked about this way back in like chapter 6? The sex chromosomes are different from each other. They're not homologous. So a female would have 2 of this one, 2 X's, but a male has 1 X and 1 Y. The Y does not have anywhere near as many genes on it as the X chromosome, which is why they're non-homologous and why they're so different in shape. So at least chromosomally, this appears to be a normal, healthy male. All right, so specifics relating to chromosomal maps, something that we should also talk about. So there are 5 human chromosomes that have what we call satellites. Now, if you look at the picture on the bottom, let's see, So the white circles represent the centromeres. You can see sometimes the centromere is all the way at the top, sometimes it's very close to the top, sometimes it's in the middle. We call the top arms the short arms, and then the bottom ones the long arms. But the satellites, if you notice here, we have this thin section hanging off of it, and then a clump of DNA at the ends. So it kind of looks like little eye stalks for an alien or something. But we have found these satellites on chromosome pairs 13, 14, 15, 21, and 22. And what we've done when we studied them is we found that they carry these repeated genes that help to encode different ribosomal parts. Now remember, ribosomes are made up of RNA, ribosomal RNA, as well as certain proteins. So these carry the genes that help to create those different components. There's something that we're going to talk about later called translocation, and this is a result in exchanges in material between 2 chromosomes. Now, this is not talking about crossing over that occurs in meiosis, specifically prophase 1, where 2 homologous chromosomes exchange the same information. A translocation is looking at exchanges in information that could occur between pseudogenes and genes, or even between non-homologous chromosomes. All right, and now let's move on to section 2, detecting chromosomes. Woo! All right, so detecting chromosomes. Any cell with a nucleus can be used to obtain chromosomes to prepare a karyotype because, again, the cell, if it has a nucleus, it has the genetic material, the DNA. Now, the DNA is going to be in one of two forms, either the chromatin form, where it's all super condensed down together, or the chromosome form, where it's kind of sectioned off and chunked off. Because of this, generally speaking, when you want to make a karyotype, you're going to look for a cell going through mitosis because you don't have to worry about separating the chromatin. It's already been separated in the form of chromosomes. A lot of times prophase or late prophase or metaphase are best because then you can still get the sister chromatids together, and that makes it easy to tell where the centromeres are located. So let's see. So fetal karyotypes. Fetal karyotypes are constructed from cells obtained by amniocentesis, chorionic villus sampling, and blood cell sorting from maternal blood. So the three ways that they can get, let's say a woman is pregnant and they want to check to make sure that chromosomally there are no abnormalities to worry about. Like I said, a lot of chromosomal mutations can be very serious and even fatal. So sometimes, especially if a woman is high risk, they might do one of these methods in order to check the karyotype of her offspring. So an amniocentesis is when they take a long needle and they actually inject it into mom's uterus, take out some of the amnio fluid, and baby cells are swimming through that, so they can easily get a sample there. Chorionic villus sampling, if you remember from way back, chapter 3, the chorionic villi are these little tissue projections that help connect the early embryo with mom's uterus and what would end up becoming the placenta. So you can take a little piece of that off in early pregnancy in order to get a sample of baby's cells with baby DNA. Or fetal cell sorting from maternal blood. So remember, the placenta is an exchange port, pretty much, between mom and baby. So mom breathes in oxygen, goes through her blood, through the placenta, into baby. Baby takes in oxygen. Baby creates carbon dioxide. Carbon dioxide goes through the placenta into mom's blood and into mom's lungs where she exhales. Same with food and waste. Mom eats food, food gets digested, goes into her blood, goes from her blood to the placenta into baby. Waste products in baby's blood goes through the placenta into mom. Mom gets rid of it. Now, it's not just gas exchange and nutrient exchange. Sometimes some of baby's blood cells can also get into, or some of baby cells can get into mom's blood. So they can take a sample of mom's blood and look for fetal cells. However, that is a little trickier because it's such a small sample, and then you got to separate baby cells from mom cells. Fetal karyotypes are prepared for patients with advanced maternal age, repeated pregnancy losses, and increased risk of a chromosomal anomaly as indicated by a maternal serum marker test or family history. So again, they're not going to do any or create a karyotype for a child just because. If there's something with mom is high risk for whatever reason, or mom's had miscarriages repeatedly in the past, it might indicate something is wrong. And could be because of, again, chromosomal abnormalities. That's what we mean by the spontaneous abortion. That's going to result in a lost pregnancy. So if the doctors are concerned about the health of the baby and the health of mom, they could do a karyotype to make sure everything appears normal. Again, this is only going to show the chromosomal map, though. Preparing cells for chromosomal observation. So to obtain chromosomes for karyotypes, Cells are halted in metaphase, broken open on a glass slide, and the chromosomes are spread over the surface of said slide. Chromosomes are stained or exposed to the fluorescent DNA probes. This helps the geneticist be able to identify them and arrange them in order of size and centromere location. So here we have the cell. Now, obviously the cell's been broken, but what they did is they took a cell in metaphase and they only want to grab one cell if possible. And they want to grab multiple cells in case the first one doesn't work out. But they're only going to open up one cell at a time because otherwise you have a whole bunch of chromosomes there and you don't know if it's supposed to have, the normal amount, too many, too few, because you got so many extra. So they'll take the cell and they will break it open. They'll actually pop the cell membrane during our metaphase, and they'll kind of spread it out on the glass, which is what you see here. These are the chromosomes. They don't normally or naturally fall into these nice little maps all organized. They have to be sorted, identified, sorted, and organized by a geneticist or a doctor or someone along those lines. So they're looking at size. They're looking at the banding pattern so they can help pair them up. And they're looking at the centromere location to help pair them up. Now, if we look at this karyotype here, we see a couple major problems. So we only have one of chromosome number 2 and one of chromosome number 4. This will definitely lead to one of those spontaneous abortions because we are missing critical information there. If too much information is missing, the cells don't know how to deal with the lack of information, and so they just stop all development and trying to figure out how to say this tactfully. They get rid of it, of the baby, the fetus, the embryo at that point. This probably would not have gotten very far past the embryonic stage because we're just simply missing too much information. With the amount of information that is missing, the cell would not be able to read it and would not be able to come together to form an actual human baby. All right, so studying chromosomes. Chromosomal shorthand summarizes the total number of chromosomes, the types of sex chromosomes, and the type of aberration present. So there's different ways that we can kind of look at this. And this is just a nice quick way to talk about the number or what's going on with the chromosomes. So we should have 23 pairs or 46 total. So 46 XY means it's a normal male. 46 XX means it's a normal female. 45 tells us that we are missing a chromosome. And the fact that there's only one X tells us that it's the second sex chromosome. And in this case, it leads to what's called Turner syndrome. 47 tells us that there's an extra chromosome, and we see it's XXY, so that means that it is an extra sex chromosome, and this leads to Klinefelter syndrome. 47 XYY leads to what's called Jacobs syndrome. 46 XY, so normal number, male, but DEL 7Q. Now, what this is telling us is that DEL is a deletion. The 7Q means that it is on the 7th chromosome and it's the long arm, so the bottom portion. So part of the bottom portion of chromosome 7 is missing. 47XX plus 21. So what this is saying is we have one extra, it is female, and it's an extra 21st chromosome. So we have an extra 21st, which leads to Down syndrome. 46 XY T79 P 21.1 Q 34.1. Yeah, that's a lot going on. So 46, we have the correct number, but there's obviously some other things going on. XY, so it is a male. Now the T stands for a translocation, and it is between chromosomes 7 and 9. So there's an exchange of information between 7 and 9. It is between the short arm of chromosome 7 at band 21.1. So remember when we were talking about those different banding colors, they can be numerically organized to help you figure out a location. So P is a short arm. So the short arm of chromosome 7 at the location of 21.1 was exchanged with Q, which is the long arm of chromosome 9 at the location of 34.1. So it's an exchange of information between chromosomes 7 and 9. And then we have 48, XXYY, which is, we have 2 extra chromosomes. We have specifically 2 extra sex chromosomes, and this leads to a male with both an extra X and an extra Y. All right, so I guess I probably didn't need to go over all that chart before, because here it is again. So again, this is just showing you how it starts off with the total number of chromosomes. Then we look at the sex chromosomes. And then if there's an extra sex chromosome, it's just going to be added right to that point. If there's a missing chromosome, typically that can only happen with the sex chromosome. So you'll see a missing X or Y. And if there's anything else, a deletion, translocation, addition, etc., that's going to be in this little notation at the end. And if it's a extra chromosome, but it's not the sex chromosome, it's going to say plus whichever chromosome number. All right, so ideogram techniques. Ideograms are maps of the distinguishing features of individual chromosomes. So remember when we were talking about those in the last example with the shorthand, we were talking about the banding locations. So that's what this is talking about, how we can actually use a map of, let's just show you the picture. It'll be better that way. So these are the chromosomes. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, and X and Y. So you'll notice that this dotted line is showing you where the centromere would be located for all of them. P's are on the top, that's the short arms. Q's are the bottom, that's the long arms. So short arms above the centromere, long arms below, short arms above, long arms below. You can see we have the satellites right there. And you also notice these numbers on the sides. Now they're away from the centromere. Think of centromere as location 0 pretty much. And then look, I'm on chromosome 16. If we said P12, then that means the 12th location, the 12th banding pattern on the short arm of 16. And then we can also have 12.2 Q 12.2, that'd be the location 12.2 banding pattern on the long arm of 16. All right, we also have something called a fish technique. No, we do not insert little fishies into your DNA. Sorry. But what FISH stands for is fluorescent in situ hybridization. And what this does is you can actually combine these chromosome-specific probes, so probes that will be attracted and attached to very particular chromosomes, and fluorescent dyes to paint the chromosomes. This can allow us to label specific gene loci or locations, so we can figure out where the gene is located, how it divides, how it moves, all that fun, great stuff. All right, and that takes us to the end of this section. So if you have questions, concerns, you don't quite know what's going on, then make sure to hit me up in our Google Classroom, send me an email. Other than that, I hope this was informative and educational, and just know that I'm thinking of everyone, and I hope you guys are staying awesome and safe. All right, guys. Take care. If you have questions, let me know. All right? All right. Bye-bye then. Take care.